Well, today we're going to get into the next topic. So in the previous two lectures, we covered clinical trial. Basically, I tried to describe what is a randomized controlled double-blind clinical trial. There's three features: randomization, uh, controlled, and double-blind. So that is an individual clinical trial. Now, how does a trial play in this whole drug development process? And here we're going to talk about this. I would call it a CDP, clinical development plan. So, how do we plan to develop a new drug? So, we try to explain the development and execution of a clinical drug program and issues, considerations, and steps involved. So, before I start, I want to talk about drug label. Now, the drug label is that very thin sheet that placed between the when you try to get drugs from pharmacy, you would have a paper box and there's a bottle within the paper box, and there's a very thin sheet of paper stacked in between the bottle and the paper box. And that very thin sheet of paper, if you can unfold it, it's printed two-sided, it's very thin, you can almost see through, the font size is very small. That is your drug label. So I do want to spend some time about the drug label because that is the most important document when you develop a new drug. Now, we all understand that drug development can be very expensive. Companies spend billions of dollars to develop a new drug. In the amount of money invested in developing a new drug in today's setting, it could go up to $2 to $3 billion. Be like in boy, $2 to $3 billion to develop a new drug. The reason that we have to spend a lot of money on developing a new drug is that most drugs failed. So in other words, we go through the entire drug development process, starting from preclinical, we get into clinical, phase 1, phase 2, phase 3. And at every stage of drug development, if the drug is not good, they fail. And so there's a lot of investment on failed or non-successful drug candidates. So we spent billions of dollars to develop a new drug. Now, if you and I go to buy things, we spend money, we go to the store, we pay the money, we get the stuff we want. Now, for drug companies, pharmaceutical industry, they invest billions of dollars. What do they buy? In fact, all these money are invested to buy data. Again, today we talk about data, all kind of data, big data, real world data, all kinds of data. But clinical trial data can be very expensive. So in a way, if you divide, say, $2 billion by the number of data points that you collected throughout the entire drug development process, you can see each piece of data is expensive. Now, so how do we make the best use of these data? So you organize the data. You can look at the entire drug, or we call it new drug application process, the entire documentation of the entire drug, starting from preclinical until phase 3, and you look at the entire data set and you try to consider how you organize these data, how you make sense of this data. You can think of it as a pyramid. So on the bottom of the pyramid are all your raw data. And then moving up from raw data are your summary tables, and then your study reports, and then you're going to have combined analysis. We call it integrated analysis, and these reports. And on the top of the pyramid is your drug label. So that drug label is really the summary of all the data you collected. And I would consider a drug label is the most important document when you're developing a new drug. Now, drug label does not exist if the drug is not approved. So when FDA look at your submission, all right, we call it NDA, new drug application. These are what we typically call for chemical entities. If you have a new chemical entity, NCE, and you try to develop this NCE into a new drug, and then this this process of developing and summarizing all these results, you put it together and submit to FDA, you call it NDA, new drug application. And there's another type of drugs we call biologics, or we call it NBE, new biologics entity. So typically biologics are large molecules, chemicals are small molecules. In a way, NCE you can synthesize in the lab, but I mean for chemical compound you can synthesize in the lab from the chemical lab or whatever. Now NBE biologics are mostly part of a live body, and it could be a piece of protein, or of course most of them nowadays are antibodies, and it is almost, well, I wouldn't say impossible. It's very difficult to synthesize in the lab. But what you can do with biologics is that you use cell lines to culture, so you get these large molecules. So when you put the large molecules together, you want to submit to FDA. It's called BLA, Biologics License Application. So either NDA, New Drug Application, or BLA, Biologics License Application, you're putting all these data you collected together, and you write reports, you write summary reports, you write combined reports, you put all of these stuff together as a package of document to submit it to FDA, Food and Drug Administration, for them to review and approve this drug. Now, so when you put the NDA or BLA together, you submit it to FDA. FDA will go through their review process. After their review, if FDA decides not to approve this drug, then there will never be a label for that drug. Again, I want to emphasize most of the drugs failed. Starting from discovery, we call it preclinical discovery, preclinical development, and then we get into clinical development and clinical phase 1, phase 2, phase 3, all these development. You can think of it as a weeding out process. In other words, the new drugs, if they're not good, you don't move forward. So a drug can fail at every stage, and sort of a drug candidate can fail at every stage. Only the most successful drugs, or the best drugs that you're developing, that could get to FDA or get NDA or BLA. And then even in that process, FDA may not approve this drug. So in other words, all these failed drug candidates, they don't have a drug label. Now, drug label appears at the time when the FDA claims the drug is approved. So before the drug is approved, we always have a draft label. So this draft label is called a target product profile. So I want to start looking at the drug label to say that is the guiding document of developing a drug. So again, this lecture and the next lecture, these two lectures, I'm trying to cover this CDP, clinical development plan. So how do you plan to develop this drug, and what are the clinical steps you have to go through in developing this new drug. And that's CDP. The entire CDP is guided by a draft drug label. So what you do in new drug development process is that you're looking at the target, which is you want to develop the drug into, and eventually you want to come up with an NDA or BLA to get this document to FDA. And so there's always this draft label throughout the development process. When you file an NDA, in the NDA, there's a draft label also. Now, when FDA decides to agree to approve this drug, then they will initiate this label negotiation. So this label negotiation being FDA will say, we look at your draft label, we're going to approve this drug. We look at your draft label, but you cannot say things this way. You have to say things the other way. And typically the label negotiation happens on the day before the drug is approved. So in the label negotiation, the company and FDA debate on almost every word in the label. So we can imagine that a company spends billions of dollars to develop this drug, and all the drug property are going to be summarized in this drug label. And this drug label will help the company to market this drug. So in a way, when a company come up with a draft label or target product profile, this draft label has to serve two purposes. The first purpose is that this label will help the drug to be approvable. So when FDA reviewed your NDA, reviewed your drug label, they say, yes, the drug can be approved based on its efficacy and safety and other findings. We can approve this drug. So when you prepare the draft label, when you prepare this product profile, it has to reflect the features that are going to be approvable. The second purpose of the drug label is marketable. So when the drug is approved, the company wants to market this drug. How do they market? They will have to have all kinds of commercial effort, and they have to position this drug and everything like that. And then for the salesman, for the drug salesman, when they go to the doctor's office, they'll try to convince the doctor to prescribe this drug. At this point in time, this communication, this conversation between the company's salesman and the doctor, all the salesman can say, do not go beyond drug label. So in other words, you want your label to reflect the best property of your drug. And many times, for this new drug to come on market, there may have already been many competitors already on the market. So now your new drug will be a new member to treat this condition, and you have to demonstrate your drug's competitive advantage as to compare to other drugs already on the market. How can you bring the best part of your drug to the doctor so that you can convince the doctor will prescribe your drug for the next patient with this condition? Now, in that conversation, the salesman can say anything about the drug, but everything the salesman can say, do not go beyond the drug label. So label is absolutely critical from both drug development point of view and from drug marketing point of view. So when you prepare a target product profile, this target product profile will have to achieve two purposes. One is approvable, the other is marketable. So this is a guiding document of developing a new drug. So when do you prepare this draft label, this TPP, target product profile? This target product profile has to be prepared even before the drug get into clinical development. So I'm going to talk about CDP today, clinical development plan, and I will have to start from a draft label, and we will look at how this draft label will guide this entire development process. So I talk about drug label, target product profile, TPP, and we're going to talk about clinical development plan, CDP. So how do we develop such a plan? And then we're going to talk about phase 1, phase 2, phase 3, and phase 4, including scientific principles guiding drug development. And then how do we execute such a plan? So like I said, drug label actually is a communication tool. So now, communications, there are usually multiple parties involved in a communication, all right? So what does a drug label communicate, all right? The drug label tries to communicate between two sides. One side is the company and FDA, and the other side is a physician and the patients. So it's drug developer and drug approver to communicate with drug user. All right. So how can we use this label to educate the physicians how to prescribe this drug? And how do we educate patients how to use this drug? Okay. So drug label actually provide prescribing information for physicians and providing the information and the education to help the patients understand how to use it. All right. Like I said, the label usually is that sheet of thin piece of paper in between the paper box and drug bottle. So in US, we call it package insert because it's inserted in the package. So when people talk about USPI, USPI, that is package insert, that is drug label. In Canada, they call it product monograph, all right, PM, product monograph. In Europe, they call it SMPC, Summary of Product Characteristics, SMPC. So that's the same thing, USPI, product monograph, or SMPC, they're drug label, all right? Now, like I said, every word on the label has to be agreed between the sponsor and agency. So here, when I say sponsor, that's drug company, because it's a company that sponsors the clinical trials. So when people talk about sponsor, that reflects either the pharmaceutical company or the biotech industry or whoever is sort of investing in these drug research and drug development. So every word on the label has to be agreed between the sponsor and the agency. So in this label negotiation, like I said, it happened the day before approval. The company would try to push the envelope to say the best they can say about this drug. And the FDA, which serves as a gatekeeper, will have to say, you can only say it this way, you cannot say it that way. So all these discussion, debate happens the day before drug approval. And both parties, sponsor and agency, pay a lot of attention to every word in the label. Now, another way to look at this label is that from a company's point of view, global companies like Pfizer, Eli Lilly, Bristol Myers Squibb, Johnson & Johnson, these global companies, what they would hope is that they would hope to come up with a drug label that applies globally, internationally, all right? But it never happened that way because you have to negotiate the label with each regular agency and with each country on an individual basis. So for the same drug, and the company bring up with this label, they negotiate with US FDA, they came up with the final label. And then they negotiate with another country to come up with another label. So typically, the labels at different countries, they're slightly different. But the main, I think the main information is about the same, but it's just speaking it differently. All right. So the label has to be agreed between sponsor and agency. And before this label is called label, is the target product profile, which is equivalent to the draft label. And typically, we have to pay attention about competitors in the market. So that's why the wording in the label are very, very critical. How can we help the drug salesperson to communicate with the physician as to the competitive advantage of this new drug? So a product profile, which is a draft label that is before you develop this drug or before this drug even move into clinical development, we have to come up with this target product profile. This target product profile has to have these two features. Critical features is more about approvability, or clinical success factors, like quantitative results of the drug must achieve for first human use, first efficacy dose, registration, and marketing. So these are critical features, more about this approvability. And desired features changes over time as more is learned about this compound and marketplace. So it's more about the marketability. All right. So a target product profile will have to serve both purposes, approvable and marketable. So we talk about label. Now we get into CDP, clinical development plan. What is the definition of a clinical product development plan? All right. A CDP is like a blueprint for drug development, which describes the draft package insert with the draft label, trials, timeline, resources, and budget needed for registration filing. So before getting into the details, let me show you what a very high-level summary of a CDP would look like. Let's take a look at this draft Gantt chart. All right. Here, we're looking at probably a 10 to 12 year development plan. Okay. Now, if today is 2021, all right, and we're looking at we're planning for this drug to be approved in maybe 2032 or something like that, 2031, 2032, something like that. So if we're talking about the first quarter or the second quarter of 2021, all right, so we're envisioning in the second quarter of 2021, we're going to engage in a 4-week toxicology study in dogs. Okay, so we're looking at, so here is second quarter 2021, third quarter, fourth quarter, first quarter 2022, second quarter 2022, and so on and so forth. 2022, 2023, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33. So by about 2033, maybe phase 3 will be completed. And so we will file NDA maybe 2023 or 2024, sometime around this time frame. So we're looking at about a 12-year plan from now, from where we are today. Okay. So we look at the drug, we say, for this year, pretty much we have to look at the toxicology studies. We'll make sure that the drug will have sufficient information to help us get into clinical development. All right. Now, this is the time the drug get into clinical. All right. So we see phase 1, SRD, and I will explain these terms, SRD, MRD, and things like that. All right. So right around this time, so this is 2021, 2022, about third or fourth quarter, the drug will move into clinical. Okay. So in other words, today we're planning for this drug to be developed for the next 12 years. All right. And so we have to start with the toxicology studies, and we will have to also look at different drug supplies and drug products and things like that. All right. So if we're talking about third or fourth quarter of 2022, the drug will get into clinical, then all the activities before the drug gets into clinical is what we call preclinical stage. So in general, preclinical stage, we have discovery, drug discovery. That means mainly the pharmacology studies, including animal studies or lab studies, in vivo, in vitro, where in vitro being outside of live body, in vivo being inside live body. So you probably start with some kind of tissue or some kind of you can look at cells, you can look at molecules, and all these kind of discovery effort. So what we can say is that when we talk about R&D, research and development, all right, in drug research, it's really about discovery, coming up with new compounds. So we can sort of separate between research and development, sort of in drug research and drug development, in a way that the discovery group are those scientists who are engaged in drug discovery. Their job is to keep coming up with new drugs, all right, either new chemical entities or new biological entities. And once they come up with these new drugs, in different companies, they have different names. Typically, they call it startup development. So for a drug from the discovery or the research group, in order to make it to development, is a huge achievement, because now the company decided to invest in this compound or this biologic. So then after this, I call it drug candidate, whether it's a compound or biologics, after this candidate engaged in SOD, start of development. Now, this is a stage of understanding this new drug candidate. All right. So before SOD, before start of development, the drug is under the discovery lab. So the scientist is supposed to come up with either chemical compound or biologics that hopefully it will achieve to be a successful drug. All right. So the discovery group's job is to keep coming up with new drugs. On the development side, it's different. The development side, they're not coming up with anything new, but they're trying to observe the drug or the candidate that was discovered from this research group, and they'll try to select only the best one to move forward. So in a way, you can look at the entire development process, both preclinical development and clinical development, are trying to weed out the drugs that are not good enough. So let me start out with an example. All right. When we introduce about clinical trials, we know this whole randomized controlled double-blind clinical trial starts in the 1960s and 1970s. So the key is 1962. In 1962 is the Kefauver-Harris Amendment, and they would require FDA, before they approve the drugs, they have to look at the clinical evidence of both efficacy and safety. Now, from 1962, of course, the companies started developing new drugs and things like that. The first wave of new drugs got approved are in the 70s. And the first wave of the drugs are mostly for lowering blood pressure or reducing pain. Right. So if we look at lowering blood pressure, as we know, when you measure blood pressure, there's three numbers. There's systolic blood pressure, diastolic blood pressure, and pulse rate. All right, or heart rate. Now, within your heart, there are four chambers. Well, we know that heart is about the size of your fist. It's in your chest, and it's a little towards the left part of your chest. And that heart has four chambers. their left ventricle, right ventricle, and left atrium, and left atrium. There's these 4 chambers. So what your heart does is it is pumping. All right. Now your heart starts pumping while you're still in your mother's womb. It's been pumping. All right, and will never stop until you die. So typically, I mean, if you're talking about 60 bits a minute, then it's just constantly pumping ever since the heart is in shape while you're in your mother's womb. All right, so it's keep pumping. So these 4 chambers, the heart muscles will squeeze the blood out from the heart, and so your blood gets to circulate, and after circulation, you'll come back to the heart. All right, so the heart does this. They constrict and they relax. They constrict and they relax. So the heart muscles just keep on doing these activities. Now, there are 2 circulations. There's pulmonary circulation and there's the general circulation. All right, the pulmonary circulation is when you pump the blood with CO2, or we can call it dirty blood. All right, the blood carries CO2. They go to your lung. So when your heart muscle constrict, it squeezes the blood from your heart to your lung. Right, it squeezes the blood with CO2 to your lung, and in your lung, these blood will exchange. They'll get rid of the CO2 and they'll bring oxygen in. So these are the clean blood. All right. So they bring the oxygen into these blood cells, and now it returns to your heart. And when your heart squeezes, these clean blood will go to your body. So it's constantly, every pump of your heart is squeezing out the blood carrying CO2 to your lung, and is pumping out the clean blood that carries oxygen to your body. All right, so it's constantly circulating. Now, in the 60s, when we're trying to find a drug, hopefully that will lower blood pressure, they're trying to see if they can sort of reduce the amount of pressure on your heart muscle. And the scientists found out there is a beta agonist in your heart muscle. So if they can come up with a drug to reduce the activity of beta agonist, then hopefully they can reduce your systolic blood pressure. So they came up with this drug they call beta blocker. All right, so that's pretty much the first generation of the new drugs in treating disease or conditions for patients. All right, now, so back in the 60s, the scientists went to the discovery lab, and they said, if we can come up with a beta blocker, and hopefully eventually the patients taking beta blocker could help reduce their systolic blood pressure. Now, I want to also mention about, since we're talking about beta blocker, we can also talk about the alpha blocker, which is when your heart muscle relaxes, your heart trying to bring the blood back to the heart. So remember, the heart is going through a circulation. The clean blood with oxygen will go through your body. And while these clean blood going through your body to each of your tissue and to all the cell level kind of stuff, they're not only supplying oxygen to your tissue, but they're also bringing the CO2 from your tissue back to the blood. So these kind of blood carrying CO2 will come back to your heart. And then these blood carrying CO2 will go through your lung. It'll exchange to bring oxygen in back to your heart, and then from your heart to your tissue. So that completes a full circulation. Now, how does the heart bring the blood back to the heart? Of course, the heart muscle will have to relax so that it will suck the blood back, the dirty blood from your body and the clean blood from your lung back to the heart. But that pressure is not sufficient. So your vessels will also have to have the pressure to squeeze the blood from the vessels, from the tissues, back to your heart. So if you want to control the diastolic blood pressure, so systolic blood pressure is a blood pressure when your heart squeezes the blood out. Diastolic blood pressure is when your heart relaxes and brings the blood back to your heart. And at that time, you need to have the vessels to squeeze the blood back. And then if you want to lower diastolic blood pressure, you have to find the tissues or the organs or whatever around your vessels. How can that help to relax the diastolic blood pressure? So they found in the vessels, there are alpha agonists. So that's why when you try to lower the diastolic blood pressure, you take alpha blocker. When you try to lower the systolic blood pressure, you try to take beta blocker. So the first generation of drugs to lower systolic blood pressure are beta blockers. So what they did in the discovery lab is that they look at these beta agonists. They can get it either from tissues or from animals or from whatever. They got these beta agonists. And then they put in the petri dish. And then they will try to add compound to see if this compound could reduce the activity of the beta agonist. So once they can find this compound, they call it beta blocker, and then they start developing it. If this potential candidate is successful, then they will move into development, and then they get into preclinical development. In the preclinical development, there are many different activities, but I want to mention a few activities that are very relevant to the later development in the clinical part. All right. Tox is one of the major activities. All right. So you want to study to see, does a drug cause any problems? So the toxicology department in preclinical development is very critical. For every drug, before they move into human, before they get into clinical, you have to see what potential problems the drug may deliver to any animals or to eventually the human being. So in the toxicology studies, they need to look at what are the potential side effects or adverse events this new drug or this drug candidate could bring. But also they need to identify some dosage, that they have to establish something that below this dosage is very safe. And this dosage is they call NOAEL, non-observed adverse event level, NOAEL. So the toxicology study is very important for them to establish a NOAEL from the discovery lab. So tox is a big part of preclinical study. Another big part of preclinical study is animal PK. So we will talk about PK, which is pharmacokinetics, in later slides. And PK really reflect how does your body work on the drug. So same thing with animals. So animal will sort of metabolize the drug also. So there's animal PK. That's also within the preclinical stage. And another big one, preclinical activity, is what we call formulation. All right. Now, for new chemical entity, for NCE, you come up with this chemical compound. And this compound itself may not be stable. Remember, if you want to come up with an oral drug being you take it by mouth, all right? If you come up with an oral drug, once the drug goes into your body, it will go through your GI system, it will go through the liver, it will go through the different parts within your system. and your body will try to break down this this chemical compound. Your body will act on it. So that's kind of PK. All right. But also this compound, how can you make sure it will be stable when it goes through your GI system? So usually when you formulate a compound, you need to get not only this chemical, but you will need to add like starch, other ingredients and all the stuff, and you have to process, either make it a tablet or make it a capsule or make it a liquid or patch or whatever. You have to formulate the drug so that the drug will go into your body in whatever way it should be. So drug formulation is a big part of preclinical activities also. All right. So when we say CTS, which is clinical trial supply, what is clinical trial supply? That are the drugs. All right. So you need to formulate the drug in a way that when you go through your phase 1 studies, all right, CTS, which is clinical trial supply for phase 1 SRD and MRD, all right, you have to go through these processes to come up with the drugs that's being formulated and ready for clinical use, all right? So that's why you have to have sufficient drug supply before you start with your first phase 1 study. Now, after you have these drugs, you have the tox studies, and you have some of the basic information, you move into phase 1, all right? So there are 4 phases in clinical development. There's phase 1, phase 2, phase 3, and phase 4. Phase 4 is typically post-marketing, all right? Now, the differences between these, or among these 3 phases, phase 1, phase 2, phase 3, let me try to describe it from a helicopter view, all right? So as to the grand scheme of things, why do we have to divide the clinical development into phase 1, phase 2, phase 3, phase 4, all right? Now, phase 4 is post-marketing after the drug is approved, all right? So if we submit NDA in 2032, maybe it'll be approved in 2033, and after 2033, the further development will be phase 4 development, all right? Now, the 3 phases of clinical development can be viewed this way, all right? It's all about finding the right dosage for your drug, all right? Now, for any drug, if it works, the only reason that the drug is efficacious is because this drug changed your biological system. Now, if the drug changes your system, it has to be toxic, all right? So we know all drugs are toxic because the drugs change your system. If the drug does not change your system, what it is placebo, right? Placebo doesn't change any of your biological system. But if the drug works, that has to change your system. If it changes your system, it's toxic. So we know all drugs are toxic. Now, how come there's so many drugs already approved by FDA? Because every drug, there's a non-toxic dose. All right. So in a way, you can view the entire drug development process is about finding the right dose. So how do you find the right dose? That's why we have the phase 1, phase 2, and phase 3, the 3 stages. All right. Phase 1, we started with very low dose. Okay. That's why I mentioned NOAEL is very important. All right. The tox study, the toxicology group, they will have to come up with a dose that is so low, it does not cause any problem. And in the animal studies, so how do you translate these animal doses into human doses? All right. In the animal studies, they'll try to look at milligram per kilogram of body weight. All right. Now, if you look at a mouse, you look at a rabbit, or you look at guinea pig, you look at their body weight, and you try to come up with a dosage appropriate for their body weight. And then when you translate into human body, you do the milligram per kilogram translation. In some drugs, they look at milligram per meter squared. They look at the body surface area kind of translation. But anyhow, you translate into human. The NOAEL, N-O-A-E-L, being non-observed adverse event level from a milligram per kilogram dosage or concentration, you translate into human. And in your first phase 1 study, you want to have that dose even multiplied by a factor of 1/10 or 1/20, so that so low, you want to be sure it does not cause any problem to human. And phase 1, you're looking at, when we look at SRD being single rising dose, MRD being multiple rising dose, is the doses are going to be rising. So you start with a few subjects at very low dose, about NOAEL or something like that, and you rise the dose. You have another group of subjects, you increase the dose, and have another group of subjects increase dose until you find any safety problems. If there's no safety problem, you just keep rising it. All right. So phase 1, the most important part of phase 1 is they have to deliver what we call PK, pharmacokinetics, which I will go into more detail in future lectures. And they try to achieve a maximum tolerable dose, MTD. All right. So the two major objectives of phase 1 are PK, human PK now, because we're in clinical trial, we're running trials in human being. All right. So this preclinical clinical is a very critical stage. Before clinical, you're doing all the non-human studies. It can be in vivo, in vitro, it can be chemical, it can be engineering, can be all these kind of stuff, but it's not a human being. Once you're a human being, that's clinical, right? So clinical trial is performing experiments in human body, in live human body. So that's clinical trials, all right? So phase 1 clinical trial is about rising the dose from the NOAEL, from the very low level up to MTD, maximum tolerable dose. In phase 2, you try to lower the dose from the maximum tolerable dose. You try to go down to minimum effective dose. All right. So phase 1, you try to increase the dose, and phase 2, you try to decrease the dose. Okay. So this is also phase 2. All right. Now, based on your information from phase 1, you can establish an upper limit of your dose range, which is a range of doses you cannot go beyond, because go beyond, people cannot tolerate. So that's why they call it maximum tolerable dose. But phase 2, you're trying to find the lower limit of the dose range, minimum effective dose, because below the minimum effective dose, the drug does not work. Now, if your phase 1 and phase 2 can successfully identify this dose range that is not too toxic and it will be efficacious, then you select one or few doses you engage into phase 3. So generally speaking, people will say phase 1, phase 2 trials are exploratory stages. They are trying to explore your dose range. And they call phase 3 being confirmatory trial, right? Because in phase 3, typically phase 3 are large-scale, long-term trials. That's why it takes 1, 2, 3, 4, 5 years to complete your phase 3 trials. Because in phase 3, you want to confirm whatever you found before phase 3 to establish the efficacy and safety so that FDA could approve your drug. So that's a major sort of differentiation of the phases. Phase 1, you escalate from the lower dose up to the higher dose to MTD, maximum tolerable dose. And phase 2, you try to lower the doses from MTD down to minimum effective dose. Once phase 1 and phase 2 can successfully establish the range of dose that within this range is both efficacious and safe, then you engage in phase 3 for long-term, large-scale confirmatory trials. I think I may stop here. And our next lecture will start from this slide because I haven't completed all the details about this slide. And we can go into this clinical development process. All right, phase 1, phase 2, phase 3, and what are the considerations of each phase when you design this trial. So today what we did is I will start with drug label. The draft label is your target product profile. So you start from the draft label. At this stage, you already have your drug label, your draft label. You're looking at that label. At this stage, you have to have a label. So you use this label to negotiate with FDA, and hopefully the drug can be approved, and there will be a label that the drug need to deliver. So you use a label to communicate with physicians and patients so that they understand how to prescribe and how to use this drug. And that drug label or draft label started from very beginning, and you go through maybe 10 to 12 years of process to develop this drug. Thank you.